Hi everyone, so in this video I'm going to talk about apoptosis. So what is apoptosis? It's the cell programming for death. So the cell is going to be programmed to death if it faces any kind of stress or any kind of DNA damages or any kind of abnormal thing. So this is basically the cell would be facing something really abnormal. So instead of going through a cell cycle or anything like that, or like making the cell to function normally, it just chooses to kill itself. This is why it's also called suicide or cell murder. So it kills itself so that this cell doesn't go through continuous kind of cycles, and this would lead to cancer. So to get away from all of this, the cell would just kill itself. And this step is really so important for the cells. Even in the cell development, it's really important because if you look at your fingers, it's this is how it looks like. And if apoptosis is not there, your fingers are going to be looking like this. So it's going to be like whipped fingers. What apoptosis does, it will just come here and keeps degrading all of the cells right here and makes it look normal. Also, apoptosis is really so important for the T cell and B cell formation because specifically at the T cell. So at the T cell, there are some after like recombination of the gene to make the immunoglobulin. It has a lot of defects in it. So it would have like some abnormalities with the T cells. And if these T cells would just go out in the bloodstream, it will cause a lot of problem. So what happens is that there is the apoptosis that will occur in these in order to help the cell like clean or the body to clean all of the bad stuff. So this is how important apoptosis are. So for apoptosis, we're having apoptosis. I want to talk about apoptosis in contrast with necrosis. So there is some differences here. Both of them, they trigger the cell death, but in a different way. So for necrosis, it could be because of a disease or an injury that had happened. And sometimes it goes through inflammatory responses, and it causes a lot of problem after that. So, and it could actually lead to a disease or amputation of any of the body parts because of the excessive amount of it. But for apoptosis, it's completely normal, and it happens because of cell stress, and it doesn't cause any kind of inflammatory response. So, and when you see it under a microscope, you're gonna see that apoptosis forms like blood bodies. But so it's something like that. So if we're having, this is our normal cell. So apoptotic cell would look like this. So it would form like apoptotic bodies. So it will detach from the cell, and then it will actually go. After that, it's getting phagocytosis. So it's degraded in the blood. And for necrosis, if you look at under the microscope, you're gonna see that it's like a cell that is actually getting the content of the cell outside of the cell, which is completely toxic. Because as we know, in the cell there are hydrolytic acids, a lot of enzymes, a lot of proteins. It cannot just exceed the cell like that. So in necrosis, it's a little bit a problematic effect on the body. So there are different pathways with apoptosis. But before we know the two ways of apoptosis, we're gonna know the general mechanism. So what does apoptosis actually do is that it activates something called caspases. So caspase is basically it will be able to activate some other proteases. So these proteases are like that degrade nuclear lamin. So it degrades nuclear lamin. It makes the it goes to the chromatin and it makes a fragmentation of it. So there will be a lot of fragments made by these proteases. So it goes to the cell and it keeps making it into fragments so that the phagocytosis will recognize it, and then phagocytes will come and just degrade all of that. So this is the main purpose of apoptosis. It activates caspase. So let's say that this is a signal here, the red X. This is a signal for apoptosis. So it will go and bind to a specific receptor, which is called the adaptor protein. So right now, the adaptor protein is activated. It goes and binds to the caspase. And then on the caspase, there is this part here. It's called the binding site of adaptor protein. This is the binding site. So as you can see here, the adaptor protein is bound to the binding site. So, and attached to it, this is our caspase. Okay, this is the caspase. And it's actually a dimer. It doesn't work as a, this is just a monomer. This is another monomer. So they have to be cleaved. So this is basically the site of cleavage right here. As you can see, this is the site of cleavage. I have colored it with a different color. So this is the site of cleavage. What happens is that after activation, they are both cleaved and they move to the middle so that they bind the two monomers together. So that will be our the last product here. This is the activated caspase. And then it goes to activate the other proteases to fragment all of the nucleus, the cytoplasm, all of the organelles in the cell. Every single thing in the cell is being not degraded, but it's fragmented to form the apoptotic bodies so that phagocytes recognize it and just eat it. So as I just mentioned, there are two pathways for apoptosis. The first one is intrinsic. Intrinsic meaning that the signal is within the cell itself. It's not coming from the outside. And the major contributor for that is a molecule that is in the mitochondria. So in the mitochondria, there is something called cytochrome C. Cytochrome C is basically in the intermembrane space. As you can see here, it's between the outer membrane and the inner membrane. So it's in between. It releases that cytochrome C into the cell, where it goes and binds to something called APAF1. Okay, and from there APAF1, and so this is our APAF1 here, and this is cytochrome C binding to APAF1, and then APAF1 goes through the process of assembly of apoptosome. So this is how it looks, and then from there apoptosome. Now this part here is not going to be called APAF anymore. It's going to be called this blue part will be called CARD. So what CARD does, it just goes, it acts like the binding domain to caspase 1, to caspase 9. So this is the caspase for the intrinsic pathway. Yeah, and I forgot to mention something else. So these caspases, the caspase 9, will actually activate another caspase which is called executioner caspases. And from there, it will actually go and activate the other proteases in the cell, and then fragmentation process will occur. So that for the intrinsic pathway. For the extrinsic pathway, it's basically going to be a signal from the outside. So let's say, for example, the FAS ligand, it goes, so this is basically a ligand for the death ligand. So it goes in, and this is how it looks. So this is our death ligand. It goes and binds to the receptor here, and this receptor is called fat death receptor. And attached to these, to that receptor is death domains. Those are the death domains. So these are the death domains, and these death domains will just go and bind also to death effectors. So these here are the death effectors. So right now, after the death effector is being activated, it just goes and binds to our caspase. So I'm sorry, so this death effector will go and it will bind to our caspase. This time, so this is the extrinsic pathway. It will bind to caspase 8. So this is caspase 8. This is caspase 9 here for the intrinsic. So for the extrinsic is caspase 8. So the same exact process occur. This will be cleaved and it will form our dimer here, caspase here after being cleaved, and then it will go and activate caspase executioners. These caspases will just go and also activate proteases, which then will fragment the cell, beginning from the nucleus by having the enzyme that breaks down the nuclear lamin, which is an intermediate. You just degrade that, then the nucleus won't just be able to hold itself, so it will be fragmented. Also chromatin. Yeah, so for caspase and the DNA, how it actually could fragment the DNA, it's via the inhibition of ICAD. What is ICAD? It's this part here. This is ICAD, and this is CAD. So what CAD normally does without its inhibition, so this I is for inhibition. So this part here is allosterically binding to it to inhibit the activity of it. What this does, it actually goes to the chromatin and it starts to fragment it. So what caspase does, it goes here to that complex and it separates that part and frees the CAD. So CAD right now is free and it could go and it would fragment the chromatins. And this is how it fragments the DNA, it fragments the nucleus and everything. There are still some other types of signals. They could be actually in the intrinsic pathway. It could be stimulated by an external signal. So the signal comes, but what the signal does this time, it could actually activate something called BED. So this is basically a family. It's called the BCL2 family, and this family, it has pro-apoptotic and anti-apoptotic. So some of them they support apoptosis, and some of them they don't. So what goes with the so anti-apoptotic will be BCL2 and BCLXL, and what pro-apoptotic are BAD, BAX, DAC, and PUMA. So these are the ones that are actually supporting apoptosis. So via the activation of this here, BED, it could activate the intrinsic pathway by an external signal. So right here it activates BED, and then BED would actually go and activate caspase. Which caspase it is? It's caspase 9, and then it triggers apoptosis after that. Also there is P53. This is a major transcriptional factor. It's really so important. It goes and it actually enhances the production of these proteins right here, which is pro-apoptotic, and it causes apoptosis at the end. So it's a signal. So whenever there is a DNA damage, the first signal that will happen, it will go to the P53. It will free the P53 from the M2 molecule, and it will phosphorylate it, and then it will actually be activated, and then it will go bind to the transcriptional factor and make the transcription for these proteins right here, the pro-apoptotic ones, and from there apoptosis will be triggered. And that's it.